Okay, I'm hoping this will work. So as Hannah mentioned, the title of my talk today is Changing Seas, Biodiversity Change in the Recent Past. But I'm not just going to talk about the marine world. I'm going to hopefully talk about a change in biodiversity across the entire planet. And evidently, that couldn't possibly be the work of a single person. So the work I'm going to present here today is the result of collaborations with lots of different people and has been funded by many different sources over the last few years. So science is a collective effort. So evidently, all of this is a result of the work and of lots of different people. But they're not responsible for me saying anything silly today. Anyway, so I'd like us to start this talk by doing a collective exercise. I'll ask you to close your eyes for a second and just think about a special place. And it could be anything, a place just that is special to you. It could be, I don't know, a beach that you like to go to, or a place where special events have taken place for you in your life. Because this presentation starts, you can open your eyes now. This presentation starts in a place that's really, really special for me. It's been part of my life for the most part of the past 20 years or so. It's Lizard Island in the Great Barrier Reef, where I've been going pretty much every year since 2002. So a very long time. It's a long-term study site for me, but it's also special because if you've been going to the same place for 20 years, it's sort of intertwined with my life. So it's a place where I decided to have children, where I did fieldwork while pregnant. Lots of good things and bad things have happened while I was at Lizard Island, and this reef in particular, which we call trimodal, is a place where I've been going and where I've been working since my PhD. And in my PhD, I had this kind of a crazy dream that I wanted to map a section of the reef and identify all the corals that lived within that section of the reef. And at the time, it was something that was not feasible to do because working underwater has a lot of constraints, and for example, GPS doesn't work underwater, and so mapping is harder, and it was just not. It would just be too time consuming. But and about 5 years ago, I managed to achieve what I set out to do with my PhD. So I guess it goes to show that it pays to be stubborn, and we mapped a section of the reef that may not look very big in this photograph, but it's actually about the size of a rugby pitch. And I couldn't do this by myself, evidently. We enlisted the help of a torpedo robot, which you see in the top right corner here in this slide. And this robot goes around the reef and it takes thousands of photographs. And then through these photographs go through processing pipeline to construct these orthomosaics of these photographs, and also 3D reconstructions of what the structure of the reef looks like. And this means that we can now understand how diversity is distributed on reefs in ways that was just not possible until very, very recently. And this allows us to understand what drives coexistence, what drives biodiversity on coral reefs in ways that were just not possible before. So that's super exciting. And then we spend very many hours doing this in this position. That's me, the yellow fins there, with our head in the coral, just annotating these big maps. And to produce something that looks like this, a map of every coral colony that we can see on this section of the reef that is identified to species. So each of these dots on this map is a coral colony, and different colors represent different species. And so we were doing this work at Lizard Island, but we were interrupted, and I'll explain why in very shortly. But this map sort of forms the background of several lines of investigation ongoing as part of my lab. So, for example, a former PhD student, Kyle Zawada, worked on predicting demographic rates from traits, because we can keep track of coral colonies and follow them through time. So we know how much they grow, when they die, and when they appear. Another student whom I co-supervised, Damaris Torres-Poliza, whose main supervisor is Josh Maiden at the University of Hawaii, is studying how the structural complexity of the reef affects the distribution of corals. And another student, Vivi Brambilla, is looking at how the structures that corals build affect the environment, and how that in turn affects the corals as well. So lots of ongoing work that is coming from this big effort in data collection. But as I was saying before, we were interrupted. And although I'm very proud that we were able to count over 10,000 coral colonies, I was hoping we'd do at least 4 times that. But Lizard Island was hit by 2 cyclones 2 years in a row. So Cyclone Ita in 2014, and in 2015, Cyclone Nathan. Cyclone Nathan was the one that interrupted us, and we had to be evacuated from the island. It was a bit of an adventure, to be honest, in that the cyclone came from one direction, and there wasn't enough time for us to leave the island. So we had to just, tie everything down and move into the library and wait for a few days, and then the cyclone changed direction, but there was a period of a couple of days of respite, and that's when we left the island. By the time we got to the mainland, the cyclone came back and hit the study site that I just showed you, and I will show you in a bit what it looks like now. The following year, 2016, is probably, it's definitely etched in the memories of people who work on coral reefs as the largest global bleaching the world has seen. Up to 90% of the corals of the reefs in this section of the Great Barrier Reef were affected at this time, with up to 90% mortality as well. And then in 2017, there was another bleaching event that also hit this part of the world. And this means that the reefs around Lizard Island lost, in a short period of time, in a period of 4 years, about 80% of their live coral cover, and about half the species that we could find there. And this means that the beautiful reef I showed you just before now looked like this. So basically, it was no longer a coral reef. Some of the structure is still there. A lot of the structure is still there because corals are glorified slimy rock, but there's no live coral, or very little live coral. And like I said, this is my special place, and so for me it was really hard to see this happening. So hard, in fact, that I've been sitting on the data that we collected in 2014 and 2015, and I couldn't really bring myself to analyze it, because it felt like it would be a lie, in the sense that the beautiful reef is gone, it's not there anymore. So publishing about what it looked like before felt wrong, and I guess if I'm honest, just because it was so I don't know, emotionally draining to go back to working on those data. So I guess the reason why I tell you this story is because I worry about how biodiversity is changing across the planet, and I worry about it for for general reasons, but also for fairly personal reasons, because I chose to work on coral reefs because I care about biodiversity, or vice versa. I'm not sure. The two things are really intermingled. So I guess the big question that I work on is how biodiversity is changing across the entire planet, because what happened at Lizard Island is not necessarily representative of what's happening across the entire planet. And we should never mistake anecdote for data. If we want to know what's happening to biodiversity across the entire planet, we need data from across the entire planet. And so before I show you the rest of my talk, I actually wanted to ask Anna if you can do the first poll. Oh, here we go. So I'd like to sort of poll the audience to see if you think that in the past 50 years, what's happened to the number of species that live at any one place.
Okay, everyone, just a couple more seconds for you to vote. Apologies for the not changed spelling mistake.
Oh, I didn't even notice that. Okay, so the vast majority of you think that at any one location, the number of species at any one place has decreased. I'm going to stop showing that. So I don't know what I did wrong here, Hannah. Here we go. So if we want to answer that question seriously, it's not the work of just one person. We need to ask the help of lots and lots of different people who have been collecting biodiversity data across the planet. And that has been the goal of the BioTime Database, which is a database that sort of brings together the work of I think at the moment it's nearly 300 people who have been collecting the biodiversity time series across the world. This is the author list of a paper we published in 2018. The database is still growing, and it's based here in St Andrews, and it grew out of an ERC project that Anne Naguran led, and where I was a postdoc. And if you're eagle-eyed, you may have spotted a name in the middle of these 300 names, which David Patterson, so the director of MASTS, and is one of the contributors of data for the BioTime database. And so if we combine all of this data, we end up with over nearly 400 source data sets, over 12 million records across over 600,000 locations, and keeping track of 44,000 species, and spanning from the 1800s to the present. And so this is what the database looked like in 2018. It's a live resource. It's still growing, and we hope to keep it growing at least for the next couple of years. We have funding to keep it growing for the next couple of years, and maybe longer. Who knows? If you have more data and you want to be part of this, please do get in touch, and we'll add you to the BioTime consortium. And of course, if you want to use the database as well, it is public, and we very much hope that people use it. So this is what the data looks like. This map has gray dots to mark the locations of all the samples in our database, and then the colored dots are color-coded by taxon, and they are sort of located in the central location of each of the 385 studies. And every time I show this map, I always have mixed feelings, because on one hand, I'm super proud of it. I think it's pretty big, On the other hand, I see lots of empty space, and so I guess maybe I should just be happy that we're not done. There's a lot more that we can do in terms of making this a better resource. So in 2014, we published the first analysis of the BioTime database. At the time, the database was smaller. We only had 100. We only had 100 data sets. So what we did at the time is to fit a model that estimated species richness change as a function of time across the entire database. And so we allowed different slopes for the different studies, but we also estimated a global slope. And this was really surprising. So actually, I have to say, to the 71% of you who said decreased, you were wrong. On average, actually, species richness has not declined. It has not changed on average. The average, of course, is an abstract construction, and what is actually happening here is that there are lots of decreases across the planet, but there are also lots of increases, and lots of places where not a lot has changed in terms of species richness. And this was really surprising when it came out 6 years ago now. But these results have held through time, as through multiple analysis of also independent data sets. What we do find that is systematically changing is the composition of the assemblages. So this is another analysis that we did of the database at a time. And here, instead of looking at just the total number of species, as on the panel on the left, we're looking at a community similarity, comparing through time. So comparing each assemblage to what it looked like at the start of this time series. And if it hasn't changed at all, so if all the species that were present at the start are still present, then similarity is equal to 1. And if there are no species in common between the start and of the time series and the point, then the point in time, then that value is 0. And what we see is that there is a very systematic decline in similarity. And what this means is that there is a great deal of turnover in terms of the identities of the species that we see in any one location, and this turnover is higher than we would expect by chance, according to at least 2 different null models that we could use. So the surprising conclusion from our first analysis of BioTime was that we do not see evidence for systematic loss of biodiversity, but we do see evidence for systematic change in biodiversity. We also see quite a lot of variation, and I will keep sort of going back to this idea that the average is only an abstract construct, and maybe the focus on the average is misguided, and what we really need to understand is this variation. So the first attempt that we did at trying to understand this variation looked at spatial variation, and this is an analysis that we've come to call the geography of biodiversity change. It's work that we did as part of an STIV working group called S-Change, which was led by Shane Blows and Sarah Supp. So they led this paper in particular. So what we did here is a different analysis to the one where we did before. In the previous analysis, we looked at the study level, but in this analysis, we were concerned about effects of spatial scale on our conclusions, and so we standardized spatial scale across all our study sites. So we broke up, basically we gridded the planet, and then we looked at how many time series we had within each of the grid cells, and we worked at the grid cell level, but always respecting the what's the word? Always keeping the studies, the time series within studies and not across studies, because that wouldn't make sense. So this meant that we went suddenly from having hundreds of points to having tens of thousands of points, and so we started feeding models to 50,000 time series, which means every time that I came up with a slightly different analysis to do, I would ask Shane, why don't we try this other thing? And it would take multiple days to run on a computing cluster. Anyway, the approach we used this time around was not very dissimilar to the one we did before, in that it was still a hierarchical model, but now we used Bayesian models, and we looked at different predictors of the spatial distribution of change. And so this is a paper that came out late last year in Science. We mapped biodiversity change. This is a map for species richness change across the planet. And it's color-coded according to whether for that region the average in species richness change is above or below the global average. So blue is above and red is below. And what we can kind of see, the pattern that kind of emerges, is that towards the tropics, we tend to see more reds, and then towards the poles, we tend to see more blues, at least for the marine data. For the terrestrial data, the pattern is a bit more complicated. There's less obvious patterns. There's also less strong trends on average. Although I did show this map at a presentation, and someone suggested to me that it looked a little bit like a map of human distribution. So it's possible we're seeing here a signal of where humans are. So that was for species richness. If we look at turnover, which is where we see much more change. So again, whereas all of these are deviations from the global mean, but I need to point out that these are very, very small values. Whereas in turnover, we see much stronger trends happening, and a really interesting thing that we see is that here I'm showing you both the marine time series and the terrestrial time series on the bottom. And what we see is that most of the marine time series are changing faster than the global average, and most of the terrestrial time series are changing more slowly than the global average. And what this tells us is that actually the marine realm is changing much faster than the terrestrial realm. And I just realized that I forgot to do the second poll. Oh well. So the take-home message from this analysis is that the marine realm is a bit of a canary in a coal mine, very big canary, because 70% of the planet is marine, evidently. But in that we are detecting biodiversity change in the marine realm that is up to twice as fast as what we see in the terrestrial realm. And again, this is a pattern that is beginning to emerge, not just from our work, but also work, the completely independent data sets elsewhere. So having established that there is a great deal of spatial variation and there are interesting patterns happening in terms of biodiversity change, the next logical question that we had is what are the threats that are responsible for this variation? And so another piece of work that emerged out of the S-Change working group was looking at the spatial distribution of threats across both the marine and the terrestrial realm. And this is work that was led by Diana Bowler, and it was really like it grew out of an idea that Amanda Bates had during the working group, and it's a paper that came out this year in People and Nature. So what Diana did was to put together different spatial layers of anthropogenic threats across the planet, So the layers include things like climate change, human use, just the distribution of human populations, pollution, as well as the potential for invasion by alien species. And then she put together this cumulative map of where we think that threats are highest. And as you can see, this will probably come as no surprise, there's quite a lot of clustering in terms of where the threats converge. It turns out there's also some pretty interesting patterns in terms of how the spatial distributions are correlated across the marine and the terrestrial realm. In the terrestrial realm, we see it in the top here, we see that there is quite a lot of positive correlations between pretty much all of the threats, with the exception of climate change. So these are the climate change here at the bottom, and you can see that they're correlated within them, but not across with the other types of threats. But all of the other threats are very well correlated. And what that means is that for the terrestrial realm, there is this multiple jeopardy, so multiple stressors happening at the same time in the same places, and then other places are less affected. And the big exception here being climate change, in that, and this and again we kind of knew this anecdotally, in the sense that we know that places like, for example, the Arctic tundra, which is warming a lot, are very sparsely populated and don't have lots of other threats. Interestingly, what we see in the marine realm is that there is far less strong spatial correlations, except the climate change threats are more correlated among them. I mean, they're still separated from all the other threats. So these are the patterns between the two, like in terms of strong correlations. And we can also look at across all correlations, not just the positive strong ones, but across all correlations, and how they are correlated to climate change. And what we see here is that whereas in the terrestrial realm we have this very strong, well, not very strong, we have this a tendency to have negative correlations between all of the threats that are associated with the presence of humans. In the terrestrial realm, we actually see weakly but positive correlations. And what this means is that places that are most affected by climate change in the marine realm will tend to also have other threats there as well. Okay, so the next analysis that I want to talk about is a paper that was led by a former PhD student, Laura Antão, who is now a postdoc at the University of Helsinki. And Laura wanted to focus specifically on climate change and whether or not temperature change was a predictor of biodiversity change. And this is the paper that came out fairly recently in Nature Ecology and Evolution. And so for this analysis, Laura used a subset of BioTime. We needed sort of longer, slightly longer time series to have any hope of detecting a signal. So we chose the proportion of the time series that have at least 5 years of data, which gave us over 20,000 time series to play with. And then the other, when we looked at the distribution of the data that we had, because we had different predictions for what was happening in the tropics versus elsewhere, we realized that really we could only do this for the temperate regions. Again, the data was gridded for constant scale. So what Laura did was extract temperature time series for each of our time series, and then relate to the biodiversity time series. This is what the slopes of the time series, of the temperature time series look like. And you'll see there's actually quite a lot of variation. So even though we know that the planet is warming on average, it's not warming at the same rate everywhere. And actually some places, in some places, some of the time series actually corresponded to cooling, because warming is not monotonic either. So we know that, for example, here in the UK, the 80s were particularly cold. So the distribution of temperature change that we see for our time series is what we have here at the top. So there are both sort of declines in temperature as increases in temperature, but definitely on average the planet is warming. So the warming is actually faster in the terrestrial realm than it is for the marine realm, at least for the time series that we have. And we also see this variation in terms of species richness change. So there's both increases and declines. And for these time series in particular, we see a slight increase on average, although this is not statistically significant. So effectively, although the average is above zero, the confidence intervals overlap zero, so we can't detect an increase or a decrease. And the question that we had was whether or not there was a correspondence between these two curves, right? So are the things on the right the same as the things on the right on the two graphs? So Lara fitted a model of richness change as a function of temperature change, as well as and its interaction with the long-term temperature, because we felt that how warm it was to begin with might also be a predictor of the effects of warming. And what she found was that for the terrestrial realm, we really couldn't detect an effect of warming on species richness change. But in the marine realm, we did detect an interaction, and a positive interaction. And if this analysis ties your head in a knot, I know it did tie mine at the start. What this actually means is that species richness increases with warming, and it increases with warming faster in the warmest locations. So the take-home message from this analysis is that there is an effect of temperature on species richness that we detect. We detect it only for the marine locations and not for the terrestrial locations. And the warmest temperate regions seem to be increasing in species richness with warming. And if we think about this, and if we focus on the fact that we used only data from the temperate regions, it actually makes sense, and it is consistent with having species migrating towards the poles as the planet warms. And it makes sense because on average tropical locations have more species than and diversity decreases towards the poles. It makes sense that it is the warmest locations, the ones that are subtropical or closest to the tropics, the ones that see a bigger signal because they're closest to the sources of climate migrants. Okay, and I'm nearly done with my presentation, and I wanted to go full circle and take you back to where we started, to the Great Barrier Reef. Thankfully, the story doesn't end in 2017 when we had just bare rock. We have been making some progress in terms of understanding what drives biodiversity on reefs. You may or may not recognize this shape, in brown, but it's the same reefs I showed you before, and the dots are the same ones that were colored at the start of my presentation. What we've been focusing on lately is coming up with ways to describe the shape of the reef and the structure of the reef, and trying to understand how the structure of the reef determines distribution of diversity on a reef. So the work of the Maris has shown that actually with 3 variables, rugosity, fractal dimension, and height range, we are capable of describing the shape of the reefs remarkably well. And the 3 variables are so closely, they capture, they're not independent, but also capture additional information. So and they are connected into a plane, and that allows us to describe in a lot of detail what the reef, what the structure of the reef looks like. And what is really, really interesting is that with these variables, we are able to predict with some success both the species richness that we see at each location, as well as the relative, as well as the total abundance that we call of corals that we see. And just to live on a positive note, this is what the reef, these are outlines of what the reef looked like in 2014, before we had a series of disturbances affect the reef. And in 2015, there was a drastic decline in the abundance of corals. In 2017 and 2016 and 2017, the decline continued. But in 2018, things started coming back. And so we see on the reefs around, there's lots of baby corals, and this helps me understand how it is possible to experience such a big destruction of a once thriving reef, as well as not seeing declines everywhere. The story doesn't finish when something is destroyed. There is hope in that most places seem to be able to recover, even if they don't recover to what they were before. They most likely recover to something slightly different. And this is what we hope to understand and explain in the near future in my lab. Okay, just my final slide, I promise. I wanted to plug something that we're doing as part of BioTime. We really want to highlight the value of data collection for biodiversity. So we've started compiling stories of what scientists experience when they're in the field. It's an initiative called Storytime, and we publish a story each week about all kinds of crazy things that happen to people in the field. So if you do check it out, and if you'd like to contribute, get in touch with us. Okay, thank you.
Great, thank you so much, Maria, for that really great talk. And it's nice to know that obviously BioTime is still open to receiving more data sets, as you just said, and I'm sure plenty of our attendees have stories that could feature in the Storytime data set as well. So for all our attendees, this is now the Q&A session. So if you have a question that you would like to ask Maria, please pop it in the Q&A box. If you're joining us through Zoom, that is at the bottom of your screen. If you're joining us through YouTube, please pop it in just the comment section that is below as well. So we're going to jump straight in, as I can see that we've already got a couple of questions submitted. So we'll just be working through those as they come in, and we'll see how we get on. So let's start with our first question from Ricardo, asking, have you studied seasonal variability in biodiversity trends, turnover or alpha biodiversity, especially in temperate and polar areas? Should we expect important differences among seasons depending on the location?
That's a really excellent question, Ricardo. No, we have not, is the short answer to that question. So far, all of the analysis that I've showed you have worked, have an annual resolution, annual temporal resolution. We do think that there is probably really interesting things happening in terms of seasonal variation, but it's not something that we've looked at yet. So, go for it.
Great, thank you. Our second question refers to those final slides presenting the extraction of shape and how reef is changing over time, asking what method did you do to achieve this? Those figures that you showed us, these images are great, and I find the photogrammetry as a tool for monitoring a really interesting concept.
Yeah, okay, so it's a fairly technical answer that is required of this question. So my advice here would be to check out the paper on bioRxiv. So what we do is like we use photogrammetry to construct the 3D models. And then we extract the variables from the digital elevation models. I forgot to say, but actually that paper, the paper that relates to that section of the presentation is on bioRxiv, and I believe the link is going to be on YouTube when Hannah puts it up. But actually this week we just heard it's been provisionally accepted in Nature Ecology Evolution, so it should, like, the paper itself should come out as well soon.
Okay, thank you. I'm going to take a question that has come in from YouTube, and it's from David asking, is there much success in artificial coral reef recovery programs?
Oof, I am totally the wrong person to be answering that question. I know that there is a lot of effort put into coral reef recovering through artificial reefs. My understanding is that most of the success is short-lived and small scale. And so if we really want to do something that is of benefit to coral reefs, we need to go carbon neutral more than build artificial reefs.
Understood. Okay, thank you for that. Next question, coming back to Zoom, is from Ian asking, how do you square up the much stated observation that extinction rates are currently increasing globally with your observation that biodiversity has on average remained the same over the last 100 years or so? Is it just the loss of species through extinction is not occurring fast enough to significantly affect species richness on ground at individual locations?
So I think it's a combination of two things. Yes, the time scale is a really important one, and it's something that it took me actually looking at the numbers to realize is that it is the fact that we have elevated extinction rates, and that we are in the midst of a mass extinction does not mean that mass extinction is not stoppable. We need to maintain the current rates of extinction for several thousand years in order to actually see a detectable dent in the number of species that we have on a planet. So that's the first answer to that question. The second part of the answer to that question is that it's also a case of spatial scale. So whereas we were working at local scales, extinction rates relate to the global scale. And because we have about 9 million, well, we think we have around 9 million species on the planet, you can imagine lots of scenarios where we lose species globally without losing species locally if we, for example, end up with the same species everywhere. So it's a scale thing.
Mm-hmm. Okay, thank you. Next question from Althea asks, did you control for time series duration in your analyses? Starting point is likely to influence how many legacies are captured by the analysis.
Yeah, really good question, Althea. So we did and we didn't. So we did in the sense that our model, like to estimate the global averages, our models take into consideration how long a time series is. And so longer time series have a bigger pull on the global average than shorter time series. With regards to when so not just the duration, but the starting point, that's much trickier to control for. What we have done is lots of post hoc analysis where we look at the effect of start of the time series on the slope, and we can't really detect an effect. So I'm reasonably confident that the results are robust to that.
Okay, that's good to know. Thank you. Next question is from Tim asking, do your findings have any implications on how marine protected areas set targets for goals and monitoring?
So one thing that I think is important from the point of view of marine protected areas is actually is related to the I think the second last analysis that I showed, the climate change one, in that because we're finding this influx of climate migrant species, I think we need to be careful in our marine protected areas that we don't necessarily treat those as invaders, because and that also we because they're coming in likely because where they're coming from is no longer suitable, right? And so this is in some ways part of adapting to change, and so the tricky thing about protected areas is that they tend to be fixed in space, and they're created so that they protect where things are great now. And increasingly, I think people are realizing that they need to be more flexible, and then maybe it's possible that protected areas need to migrate as well to follow the things they are trying to protect.
Yes, yeah. Thank you. Next question is from Andrew asking, he says, you had a slide suggesting that big biodiversity change in the Indian Ocean, which is well away from where there are human populations, that did not seem to be driven by climate. What do you think the cause of that was?
I don't know, Andy, is the short answer to that. So work that is ongoing at the moment, led by another PhD student I co-supervised, Gorgana Daskalova, is actually looking at attributing the variation in change to different drivers. So putting together the work that Diana Bowler did, sort of using both cumulative and the different stressors, and looking at whether there is evidence of different combinations of threats producing different results, as well as whether we see evidence of antagonistic or synergistic effects of different stresses. But that's work in progress. It doesn't look like there's a simple answer to that question, and I haven't looked in sufficient detail to the Indian Ocean in particular to have an answer to that.
Okay, thank you. And we have one question left. So if any of our attendees who are still with us who have a burning question that they want to ask live today, please type it in the Q&A box, or alternatively on YouTube. The next question that we have is from Cher asking, have there been studies in variations in the detectability of biodiversity change across latitudes? Say signals are harder to detect in highly complex, noisy biomes.
So across latitude, the answer is yes. So the geography of biodiversity change analysis that showed that's the paper that came out late last year in Science, led by Shane. I had all my money on the tropics being significantly lower than everywhere else. That was my experience. So that was my my bias. That's what I was expecting to see, and we didn't see a super clear signal, and to the extent that the big signal that emerged from that analysis was the difference between the marine and the terrestrial realm, rather than the difference between the different latitudinal bands across the planet. Highly complex and noisy biomes. Again, so that analysis as well had looked at the effect of biome in predicting change, and some biomes did jump out as having faster change than others, but I can't tell you off the top of my head what they were, and if there was something like there was nothing obvious that jumped out at us in terms of which biomes had increases versus declines, and which ones had the fastest changes. I'm afraid it's just complex and nuanced, and the answer is almost always it depends.
All right, thank you very much, Maria, for going through our Q&A and for today's MASTS webinar. We don't have any more questions that have been submitted, and I can't see anything else coming in. So I think that just leaves me to say thank you very much for being today's MASTS webinar presenter. For everyone who is still with us, if you saw some slides that you would like to go back and see, or check out those links for papers that Maria has mentioned today, they will all be available on YouTube at the end of today. So those links will be in the video description, as well as this entire presentation and its Q&A. So don't worry if you missed something, it will be there very shortly. Thank you very much, Maria.
Thank you.
For all our attendees who are still with us, I would just like to highlight the rest of our MASTS webinar series that we have coming up. The 2nd of September is our very last MASTS webinar for this time, just because in October, the 5th to the 9th of October, it's going to be MASTS annual science meeting, and this is going to be online. So please stay tuned for more information about that. But in the meantime, to get your marine science fix from Scotland, please sign up to any of these talks that you see, and if they interest you, please use the same registration link that you have used before. And we hope to see you again very soon.